Thanks for the introduction. Today I'm going to talk about a project that we started in the lab where we accidentally found a highly potent regulatory doxycycline RNA aptamer, where we actually just wanted to compare Celix methods, which is quite funny. So as a start, sorry for the poor resolution, don't know why that happened. Of course, we're working on a lot of switches, as we said. For us, especially interesting is that we can engineer synthetic routing switches from a regulatory RNA aptamer coupled to an expression platform. And when you have the ligand of choice present, the regulatory RNA aptamer undergoes conformational changes, thereby affects the expression platform and can switch on or off the switch. And particularly interesting is that these routing switches are modular, so you can take whatever expression platform you're interested in, from translation, initiation sites, ribozymes, splice connects, and combine them with a regulatory RNA aptamer of choice, and create a new synthetic routing switch that reacts to your ligand of choice. But I hope you can see that. So, but how do you get your regulatory RNA aptamer? You perform Celix. And when you look at Celix methods, you either have conventional Celix, where you couple mostly the ligand to something, and then you incubate your RNA pool with this immobilized ligand, and then you elute your RNA. Or what we have established in the lab is RNA capture Celix, that we don't see, is that you immobilize the aptamer and capture oligo that is coupled to a paramagnetic bead. And then you incubate the RNA pool with the ligand of choice, and those candidates that undergo conformational changes are then eluted from the capture oligo. And we hypothesized that this is the more streamlined way to find regulatory aptamers that undergo conformational changes upon ligand binding. But to show that, we had the idea that we use a target like doxycycline that is quite promising to find a new aptamer, since we have already tetracycline that is good regulatory RNA aptamer, and also doxycycline, I think everybody has in the lab due to PET-based systems, and use one RNA library that's exactly the same for both Celixes. See that we get enriched pools, perform in vivo screening to identify regulatory sequences, and then look what the effects were. And so we started with a parallel Celix approach, and where we compared column-based Celix to capture Celix. While using the capture Celix pool due to the necessity of the docking sequence in here. And what we got is that we had a quite high enrichment in the column Celix against doxycycline in around 10, after increasing stringency by encounter selection and decreasing the elution concentration. Whereas we had similar enrichment in the capture Celix after 7 months. But still the question remained, are there any regulatory active sequences in those enriched pools? So we performed in vivo screening, and we do this mostly in yeast. So we have a yeast plasmid where we clone our enriched pool into, and then the sequences are located in front of GFP, and we have Cherry as a normalizer. And we measured those yeast then in a cytometer, and what we see is that those regulatory active aptamers work in a roadblock mechanism. So in the absence of our ligand, the premiumization complex for translation can pass through the aptamers structure, find the start codon, and will give us GFP expression. Whereas when doxycycline is present, we expect those regulatory sequences to change their stability, thereby blocking the premiumization complex from passing through, and we get decreased GFP expression. And so we then. I'm very sorry for this resolution. I have no idea why I took that. But in any case, the left part is the column-based Celix, and the red bars are the median switching factors. So what we see as change between width and the alpha ligand, and we see there's almost no switch. There's at 1, nothing happens, except at 1 point. There, I will come to it later. And then on the right side, we have capture Celix, where we have a median switching factor around 3 over the whole pool. So we really saw a very streamlined, very effective enrichment of regulatory aptamers in some way. We then sequenced those candidates, and we found out that this little point up here, which is a total outlier, is G12. This is one sequence that we found in this whole pool. So we screened thousands of candidates. We then tested, I think, 300 plates. We sequenced them all, and only out from the common Zilix came G12. It is one clone, and it had out of the box a 7-fold switching factor. Whereas more or less the whole population from capture Zilix is just one sequence. That's C1, as we call it. There was in multiple clones, and the other sequences that were switching in there are point mutations of this, and it had a 3.3-fold switching. This raises the question: what happened during Zilix? So, because I didn't expect that to be that crazy, so we did NGS analysis, and first of all, we tracked our two candidates, G12 and C1. And on the left side, for the column-based Zilix, we see that it starts that both seem to enrich in the first few rounds, then they sharply drop, and then they start to fluctuate through the selection process. None of them comes into the top 100 sequences. And even interesting is that C1 is higher in its abundance in this pool in the final round of 10 than G12, but we found G12. So the chance of finding a regulatory aptamer in those pools is very low when you do column-based Zilix. In contrary, in the capture Zilix, C1 after round 1 was already in the top 10 sequences, making up position top 6, and was the top 1 sequence in the pool from round 4 onwards until the end of the selection. In contrast to G12, that continuously de-rich in abundance in the pool, ending up not even being in the top 1,000 sequences. So we asked what happened, and we then simply tested G12 in the capture Zilix setup to hybridize to the capture oligo, and it didn't work. So this might be due to the structure that I showed you later, but it seems that due to the structure of this particular aptamer, it doesn't couple to the capture oligo anymore, and it can't be selected by capture Zilix. And with this, we asked us what happened in the pool, how this Zilix actually influenced the pool composition. And so there's a lot of motifs in there. So we did a motif search by group. We clustered them and tracked them over the course of the selection. What you should see over here in the column-based Zilix is that you have a lot of motifs fluctuating through the selection, and in the end you come up with 7 motifs. And whereas in capture Zilix, we have more or less just 3 motifs coming up in the selection, and the 2 lower ones that are in green can be assigned to C1. So we have again here indication that this single regulatory aptamer was very streamlined selected through this method. We then also tested the top 10 of the last round from the column Zilix if they bind to doxycycline, and 6 out of 10 do, but none of them were found regulatory active. We then decided to go deeper into the characterization of those aptamers, and we decided to do this with G12 due to its higher switching factor. And we utilized the characteristic of tetracycline antibiotics that's quite interesting. They're fluorescent, but they're quenched in water. So when they're bound to an aptamer, for example, they start to fluoresce again. And we did this to measure the KDs. What you should see here is that with increasing aptamer concentration in the doxycycline solution, we get increased fluorescence, and we can calculate the KD of 4.8 nanomolar with that. And we also confirmed that by ITC. And when you do the same thing with tetracycline, we have no signal at all. So this aptamer perfectly discriminates between doxycycline and tetracycline. And what you have when you have some cool characteristic like this on hand, you can do something else, which is stop-flow spectroscopy, and the pictures here work. I'm really sorry, no idea what happened. Maybe the change from Mac to Windows now. We talked about it. What you should see here from blue to gray is experimental data of the binding. So we resolve here the binding over time. We have the fluorescence signal on the y-axis and the time on the x-axis. And the lines that you should see is the data. So the best model that explained the observed data is a model where we have 2 conformations of the aptamer in equilibrium. Both can bind doxycycline, but R2 can bind doxycycline a bit better, but both can go into an intermediate state, R1 star and 2 star, and both then go over into the final bound state. And this final bound state binds doxycycline almost irreversible. So this model explains it that the binding is so good that you almost can calculate no back reaction, which is crazy. We then continued with very extensive mutational studies on the aptamer. And this is a summary of the whole studies. So in green, we can see positions that you can touch, you can exchange them, and you still have regulatory activity of the aptamer. Yellow is some limited variability, and red is no variability at all. And interesting is that you can exchange the sequence and also the length and the structure if you want them. Also this junction between P2 and P3. P2 and P3 seem to serve some structural purposes because you can exchange to great extent the sequence as long as you remain structured. So for example, P3 has to be 8 base pairs long, completely paired, and then you have perfect regulatory activity. In contrast to that, L2 and O3 you can't touch at all, except for one U here. This is you can touch, you can do whatever you want. And I would expect you could also just put their naked backbone and it wouldn't care. So maybe that's just like space. And that already gave us a hint that something with binding might be happening between L2 and L3. We did the whole studies in yeast, so we also did some 2 stem mutations for high regulation in yeast. And we saw some quite cool regulation happening in yeast with the roadblock mechanism. Then we thought, hey, let's compare this to previously discovered aptamers. And we did this again in the roadblock mechanism in yeast. And what we have on the left is tetracycline, topramycin, and neomycin in their best working forms in yeast, compared to 3 versions of the doxycycline aptamer, D12 and 10, so D12, the original version, and 10 as well. And what you can see right away is that we have high expression levels for all doxycycline aptamers. Again, in the same setup, it's just exchange the aptamer. And also it switches down to a similar or even lower level of expression to the other aptamers. And since we're here in the setup in yeast where this change in stability from a very loose unbound state to a very stable bound state counts, this tells us that there is a great change in stability in this aptamer scenery. And we then thought, okay, we should have a look into the structure and perform a shape map to first probe the structure. So on the top, we have M10 in this case with doxycycline, without doxycycline, shape map reaction, and this confirms or supports the folding prediction with the highest reactivity in the junction, the L2 and the L3 region. But also interestingly, we have almost no reaction for some parts of L2 and L3, indicating there's some structure going on that prevents them from reacting. And then we also did it with doxycycline and checked for the stabilization, so the lowered reactivity of the single nucleotide position. And again, we saw the highest stabilization occurring in L2 and L3, again hinting towards some role in binding and also in switching. With this, we thought, okay, you can see now we have this very great change in stability, seemingly. And we also have some hints that there is something going on between L2 and L3. And so we formed single molecule force spectroscopy. So in this method, we just place the aptamer between 2 beads, and you can pull on the beads and then measure how much resistance is caused by a structure in the middle. And you can also move this molecule then from a channel with ligand to a channel without ligand, so you get measurements with and without the respective ligand. What you get in the end are curves like that. So on the x-axis is the extension in nanometers, y-axis is the force in piconewton. And when we measure G12, we get this graph. So in blue, this is the extension, and in light gray, this is the relaxation. And what you see at the beginning is it is the fully folded structure that resists to the pulling, and then it suddenly pops open with 2 short stops at an intermediate state 1 and intermediate state 2 until it's completely unfolded. And if you then relax it, it takes some time, and it first gets into something like the intermediate state 1 until it then, in a very relaxed state, folds back into the fully folded state. And that tells us that the structure that it has to fold is pretty complex. Otherwise, it would easily fold back, but it doesn't. And then it gets more interesting when you add doxycycline to the system, because then you see that, first of all, the fully folded state seems to have a bit more resistance to the pulling, but this is actually just caused by this bound intermediate structure 1 that we can see here. And this creates a lot of resistance to the pulling, which is very interesting. And then it's at a certain point just pops open without even stopping at intermediate 2 and is fully unfolded. And even with doxycycline, it needs also a lot of time and relaxation to fold back. And more interesting is that we can calculate from the pulling and the distance pulled how many nucleotides have opened. And this tells us again, this bound intermediate 1 seems to be something between L2 and L3. With all this characterization, looking into the structure and everything, we then thought, okay, we go more into the application. And one thing that we did is we engineered an app design with G12, and we did this by combining our aptamer here in blue with the hammerhead ribozyme and randomizing the L2 loop so that we have N6 here. And the idea is that in the absence of our ligand, the aptazyme would be active, would cleave off, and when located in a 3' UTR of the mRNA would be cleaved away. But in the presence of our ligand, the ribozyme would be inactive, and we would, for example, get protein expression. But with an N6 tool, you have to find some interaction with the aptamer or some part of the ribozyme to make this switchable. So we used the established and published method by the small data, where you have your N6 loop, so you have your construct on the plasmid, you bring it into hex cells, and you then incubate it with without ligand, and you can map back your RNA reads to the DNA level in the cells after NGS, and thereby you can normalize the reads and then calculate the switching factor. And what we came up with is here on the x-axis, these are the counts without doxycycline, so when it's active. And on the y-axis, we have the switching factor. We have some candidates that show switching efficiencies between 15 and 18-fold, which is pretty good, because the original construct for tetracycline with the hammerhead ribozyme had, I think, in the best case, around 4.5. So we massively increased the switching efficiency. And with this, I'm at the end of the talk. I think the important part is that what we saw is that the Celix method and especially subsequent screening are extremely important for the identification of regulatory aptamers. But it also seems that although the capture Celix provided us more sequences that were switching, we still get the best candidate from the column Celix, but just by great luck. G12 has had vinegar binding affinity and specificity, and it seems that its binding motif is located between the L2 and the L3 region. And its full regulation is based on this stabilization of the bound intermediate 1 structure, which drives regulation. And we also were able to show that it has a high regulatory potential in yeast as well as in yeast. With this, I have to thank all the great students that helped on the project, a lot of great collaborators from Frankfurt, from Munich, and from Aarhus, and the whole lab that also worked on this project. And looking forward to your questions.